Assalamu alaikum, my dear students. Hope you all are fine and staying safe from the pandemic. Today we are going to start with the physiology of cardiovascular system. And this physiology of cardiovascular system, it includes various aspects of the physiology of heart as a pump. And it also includes the physiology of two main divisions of blood circulation. Now these two divisions of circulation are pulmonary circulation and systemic circulation. Keep in mind that the heart consists of two pumps which are placed in series. Two pumps mean the right and left halves of heart. Now these two pumps, they are connected by pulmonary and systemic circulation. Well, if we look at the functions of cardiovascular system, there are some primary functions and then there are some secondary functions. The primary functions, they include the distribution of nutrients and oxygen to all the body cells, and that spans also the collection of waste products and carbon dioxide from different body cells and to carry them to the excretory organs for excretion. Now the secondary functions that are subserved by the cardiovascular system, they include thermoregulation and the distribution of hormones to the target tissues, and also the delivery of antibodies, platelets and leukocytes to aid the body defense mechanism. Well, for everything, if you have an objective in your mind, then you can reach the destination easily. So our learning objectives of studying cardiovascular system, that when we complete our cardiac physiology, we have the objectives that students must know the functional anatomy of heart, physiology of the cardiac muscle. Also, you should know that cardiac muscle tissue is of two types, that is contractile and conducting type. Later we will look at the heart as a pump, and this pump action that will include the topics like cardiac cycle, cardiac output and venous return. Briefly, we will look at the ion channels which are present in the contractile cells and the receptors which are present in these contractile cells. The action potential from the contractile cells, and especially the ventricular action potential, that is a very important university question. We also need to learn that how the cardiac impulse is originated and then it spread through the cardiac muscle, Briefly, we will look at the ECG, and finally, uh, we will see that how does our cardiovascular system functions and modifies its function to achieve homeostasis in health and in disease conditions. So let's start with the characteristics of cardiac muscle cell, which I will teach you in the form of a brief comparison of cardiac muscle to skeletal and smooth muscle. Now, in this table, you can see that the characteristics of cardiac muscle, few of them resemble that of the skeletal muscle, and others have similarities with smooth muscle. All three types of muscles, skeletal, smooth and cardiac muscle, they have contractile filaments, the thick and thin filaments. Now, you have already studied that the smooth muscle, that is called smooth muscle because it does not has a specific and defined bending pattern. It does not show distinct light and dark bands, but our cardiac muscle, just like the skeletal muscle, it shows distinct sarcomeres and a bending pattern. Similarly, the transverse tubules, which are not that defined in the smooth muscle, in case of cardiac muscle, our muscle has well-defined transverse tubules, and they play a very important role in the contraction of cardiac muscle because they are in direct contact with the extracellular fluid, and the concentration of the calcium in the extracellular fluid, then it has effect on the contractility of cardiac muscle. And this feature of cardiac muscle, it resembles that of the skeletal muscle. It is different, cardiac muscle is different from the smooth muscle in regard of these transverse tubules, because in smooth muscle there are no well-defined transverse tubules, instead there are only caveoli. Again, the next point is the sarcoplasmic reticulum. You see, in skeletal muscle, the sarcoplasmic reticulum, they play a major role because they are the main source of calcium storage. But in case of cardiac muscle, the source of calcium ions, there are two sources. One is the sarcoplasmic reticulum and the other is the calcium which is present in the extracellular fluid. In fact, the amount of the extracellular calcium ions, that determines the release of the calcium ions from the sarcoplasmic reticulum. So in cardiac muscle, we have two sources of the calcium ions required for the contraction. That is, one is the sarcoplasmic reticulum and the other is the extracellular fluid calcium ion concentration. Well, the next feature, the distinguishing feature of the cardiac muscle from the skeletal muscle, is the presence of the gap junctions between cells. In skeletal muscle, you see each fiber, it is independent. There are no gap junctions between the cells. But in our cardiac muscle, there is an extensive network of the gap junctions between the cells, because of which our cardiac muscle, it acts as a network, it acts as a syncytium, and it contracts together. And then you can see some other features which are differentiating for the cardiac muscle. As I already told you that in the skeletal muscle, the only source of calcium ions which are necessary for activating the contraction, that is sarcoplasmic reticulum. But in case of cardiac muscle, there are two sources, the extracellular fluid and the sarcoplasmic reticulum. Now, just like skeletal muscle, in our cardiac muscle, the site of the calcium regulation, that the site where the calcium binds and regulate the contraction, that is the same, the troponin. And here it is different from the smooth muscle, because in that case the site of calcium regulation is the myosin filament. Then the speed of contraction, that is a very important distinguishing point for the cardiac muscle, because in the cardiac muscle the speed of contraction is always slow as compared to the skeletal muscle. Uh, because skeletal muscle, they can contract, there are two types of fibers, as you might have already studied. There are fast fibers and the slow fibers. But in case of cardiac muscle, the muscle fibers, they are slow, the contracting muscle fibers are slow. But it is also different from the smooth muscle, where the contraction, the speed of the contraction, it is very slow. Then the next contrasting point is the spontaneous production of the action potentials by the pacemaker cells. Uh, you know that in skeletal muscle there are no pacemaker cells, and it needs external stimulation to show the contraction. But in cardiac muscle, there are few specialized cells, and they are spontaneously active. It means they spontaneously discharge the impulses, which makes the cardiac muscle automatic. Since some fibers of our cardiac muscle, they act as the pacemaker cells. So our heart, it does not require external stimulation to keep beating. In fact, it keeps beating because of the presence of these pacemaker cells, few pacemaker cells, which spontaneously they discharge the active potential. Now, just like skeletal muscle, cardiac muscle has no tone, and the contrasting point is that in case of skeletal muscle, the effect of external nerve stimulation, it always results in excitation of the muscle. But in case of cardiac muscle, keep in mind that the external nerve stimulation, it can not only result in excitation but also in the inhibition of contraction. So the speed of the contraction, the strength of the contraction, that can be increased or decreased by the external effect. And this external effect, it could also be in the form of hormones. Our skeletal muscle, it has no effect on its contraction of the hormones, but our cardiac muscle, uh, both the excitability and the contraction, they are affected by different hormones. Then there is a mechanism of uh, stretch of the cell which produces the contraction, and it is only seen in the visceral smooth muscle. That when you have already been taught that the stretch relaxation that is present in the urinary bladder, so that is seen only in the smooth muscles. This phenomenon that the contraction takes place in response to the stretch of the muscle fibers, this does not occur in the cardiac muscle or the skeletal muscle. I would like you to memorize this table of the contrasting points, because you might get a question, a university question, which will ask you to describe which will ask you to compare and contrast the features of cardiac muscle from that of the skeletal and the smooth muscle.
Than any other system, the circulatory system has contributed strong imagery to people's daily language. Heart is a metaphor for love and for courage. People say they nearly had a heart attack to describe an experience of surprise or shock. Scientifically speaking, emotions are more a matter of hormones than myocardium. The heart is neither soft nor hard, but a muscular, fibrous pump. If we look at the cardiac anatomy, the circulatory system or the cardiovascular system, it consists of heart and the blood vessels. The heart's pumping action squeezes blood out of the heart, and the pressure it generates forces the blood through the blood vessels. The autonomic nervous system controls the rate of heartbeat, and one continuous layer of epithelium lines the entire circulatory system, the heart, and all the blood vessels. Well, if we size up the heart structure, the heart is shaped like a cone, and it is only about the size of your fist. It lies between your lungs, just behind, or you can say posterior to your sternum, and the tip or the apex of the cone, it points to the left. In most individuals, the heart is situated slightly to the left of center in the chest. The four hollow spaces of the heart, they are called its chambers, as you can see in this figure. The heart is divided anatomically and functionally into left and right sides. Each side has one atrium and one ventricle, each with a separate function. A membrane called the interatrial septum, it separates the atria. A membrane called the interventricular septum, that separates the ventricles. The contraction of the heart muscle pumps blood into and out of all four chambers in a rhythmic pattern. So, students, you should able to describe the structure of heart and its position within the thorax. The heart, which is a roughly cone-shaped hollow muscular organ, it is about 10 centimeters long and is about the size of the owner's fist. It weighs about 225 grams in women and is heavier in men. The heart, it lies in the thoracic cavity in the mediastinum, which is a space between the lungs. It lies obliquely, a little more to the left than the right, and presents a base above and an apex below. The heart is divided into a right and left side by the septum, which is a partition consisting of myocardium covered by endocardium. After birth, blood cannot cross the septum from one side to the other. Each side is divided by an atrioventricular valve into the upper atrium and the ventricle below. The atrioventricular valves are formed by double folds of endocardium strengthened by a little fibrous tissue. The right atrioventricular valve, which is called the tricuspid valve, it has three flaps, or you can call cusps, and the left atrioventricular valve, with an alternate name of mitral valve, it has two cusps. Flow of blood in the heart is one way. Blood enters the heart via the atria and passes into the ventricles below. The valves between the atria and ventricles open and close passively according to changes in pressure in the chambers. They open when the pressure in the atria is greater than that in the ventricles. During a ventricular systole, means during ventricular contraction, the pressure in the ventricles rises above that in the atria, and the valves, they snap shut, preventing the backward flow of blood. The valves, they are prevented from opening upwards into the atria by tendinous cords, which are called chorda tendinae, which extend from the inferior surface of the cusps to little projections of the myocardium into the ventricles covered with endothelium, which are called papillary muscles. So how does blood flows through the heart? The two largest veins of the body, the superior and inferior vena cava, they empty their contents into the right atrium. This blood passes via the right atrioventricular valve into the right ventricle, and from there is pumped into the pulmonary artery or the pulmonary trunk, whatever you call it. This is the only artery in the body which carries deoxygenated blood. The opening of the pulmonary artery, this is guarded by the pulmonary valve, which is formed by three semilunar cusps. Now this valve, it prevents the backflow of blood into the right ventricle when the ventricular muscle relaxes. After leaving the heart, the pulmonary artery divides into left and right pulmonary arteries, which carry the venous blood to the lungs, where exchange of gases takes place. Carbon dioxide is excreted and oxygen is absorbed. Now, two pulmonary veins from each lung, they carry oxygenated blood back to the left atrium. Blood then passes through the left atrioventricular valve into the left ventricle, and from there it is pumped into the aorta, which is the first artery of the general circulation. And the opening of the aorta, that is again guarded by the aortic valve, which is formed by three semilunar cusps. So from this sequence of events, it can be seen that the blood, it passes from the right to the left side of the heart via the lungs, or in other words, through the pulmonary circulation. But it should be noted that both atria, they contract at the same time, and this is followed by the simultaneous contraction of both ventricles. Also keep in mind that the muscle layer of the walls of the atria is thinner than that of the ventricles. And this is consistent with the amount of the work they do. The atria, usually assisted by gravity, pump the blood only through the atrioventricular valves into the ventricles, whereas the more powerful ventricles, they pump the blood to the lungs and around the whole body. The pulmonary trunk, it leaves the heart from the upper part of the right ventricle, and the aorta leaves from the upper part of the left ventricle. Now I can give you the summary of the functional anatomy of the heart. You see, heart, it is a muscular organ between two lungs in the mediastinum. Its mass is around, I already told you, it could range up to 330 grams, and it is of the size of a man's fist and composed of fibrous element. Then there are three layers around the heart, which are pericardium, myocardium, and endocardium. If we want to overview the cardiac muscle, then we will describe it as a striated, interdigitating, arranged spirally around the circumference of heart, where fibers, they do not divide or multiply to replace the dead tissue. You see, the mitochondria, they make 25 to 30% of total cell mass. Cardiac muscle uses free fatty acids for energy source mainly, and it has got extensive blood supply, like one capillary for each fiber. Then cardiac muscle shows end-to-end anastomosis of cells through the desmosomes, and it has gap junctions at the intercalated discs. Cardiac muscle is composed of 99% of the contractile myocardial cells and only 1% of the pacemaker and conducting tissue. So my dear, I hope that by now you have memorized the functional anatomy of the myocardial cells, which are 50 to 100 millimeter long, 10 to 20 millimeter in diameter. They form end-to-end anastomosis. They have a central nucleus. Their contractile unit is, of course, sarcomere with its A band, I band, H zone, and Z lines. Then a very important feature of the myocardial cells is the sarcoplasmic reticulum and the T tubules, which dip in at the Z lines. The filaments which are present in the myocardial cells are myosin, actin, troponin, and tropomyosin. Although in the tropomyosin, the amino acid sequence is different from that of the skeletal muscle. Cardiac muscle fibers are made up of many individual cells which are connected in series and in parallel with each another. Each cell, it may be 50 to 100 micrometer long and 20 micrometer in diameter. The cell membrane, that is called a sarcolemma, and there are large numbers of elongated mitochondria in close contact with the muscle fibrils. Cardiac muscle is striated in the same manner as in the typical skeletal muscle. It has typical myofibrils that contain actin and myosin filaments almost identical to those found in the skeletal muscle. And these cardiac muscle fibers arranged in a lattice work, with the fibers dividing, recombining, and then spreading again. 1% of the cardiac muscle cells are autorhythmic, and they have intercalated discs with the gap junctions and desmosomes. Although the sarcoplasmic reticulum is smaller than which is present in the skeletal muscle, but the mitochondria, they are abundant, which extract about 80% of the oxygen. And also the T-tubule system in the cardiac muscle, it is more well developed. The right atrium, it has got pacemaker of the heart, which are SA node and the AV nodes. And in addition to that, the right atrium, it receives venous blood via superior vena cava and inferior vena cava, and then it communicates with the right ventricle through the tricuspid valve. Now, the right side of the heart, it consists of right atrium and right ventricle. The right ventricle, it receives deoxygenated blood from the right atrium. Then from right ventricle, blood to the pulmonary artery, and then it goes to lungs. Now we move on to the left side of the heart, which of course that also consists of an atrium and a ventricle. The left atrium, it receives oxygenated blood from the lungs through the pulmonary veins, and it communicates with the left ventricle through mitral valve. Now the left ventricle, it pumps blood to different parts of the body through aorta. The two atria, they are separated by interatrial septum, and the two ventricles, they are separated by interventricular septum. And keep in mind that ventricles, they are the main pumping force. And I have already told you about the papillary muscles and the chorda tendoni. So my dear, you can see that atria, they are thin walled, and they have only two layers of muscle fibers, which is the superficial layer and the deeper layer. While the ventricles, the wall of the left ventricle is around 3 to 4 times thicker than the right ventricle. And it is made up functionally of two groups of muscle bundles, the inner and outer spiral muscles and deep constrictor layer. The inner and outer spiral muscles, when they contract, they cause the shortening of the heart from base to apex in the right ventricle. Now in right ventricle, contracts against low pressure of 25 millimeters of mercury. So when deep constrictor layer contracts, it decreases the transverse diameter of heart. And our left ventricle, it contracts against a high blood pressure of 80 to 120 millimeters of mercury. This concept is very important, that our cardiac muscle, it acts as a syncytium. The heart actually is composed of two syncytiums. The atrial syncytium, that constitutes the walls of two atria, and the ventricular syncytium, that constitutes the walls of the two ventricles. The atria are separated from the ventricles by fibrous tissue. Normally, the potentials, they are not conducted from the atrial syncytium into the ventricular syncytium directly through this fibrous tissue. Instead, they are conducted only by way of a specialized conducting system of the conductive fibers, which is the SA and AV nodal system. Students, in anatomy, while you were studying the cardiac anatomy, you have already studied that the four hollow spaces of the heart, they are called its chambers, and between the chambers are several valves that allow measured quantities of blood to flow into the chambers and keep the blood flowing in the right direction. Now, the valve names, they tell you the anatomical location or the characteristics. The two atrioventricular, or you can call them AV valves, they lie between the atrium and ventricle on each side. The bicuspid valve, it has two flaps, and the tricuspid valve, it has three flaps. The semilunar valves, they are shaped like half moons. AV valves, they are fibrous and elastic tissues. These valves are connected by chorda tendinae to the papillary muscles. If a chorda tendinae becomes ruptured, or if one of the papillary muscles they become paralyzed, the valve it bulges far backward during the ventricular contraction, results in severe or even lethal cardiac incapacity. So all these, the valves, the chorda tendinae, and the papillary muscle, they act as a single atrioventricular valve mechanism. So the function, if we describe the function of the papillary muscles, you should know that the chorda tendinae, they are attached to the papillary muscle, and the papillary muscles, they contract when the ventricular walls contract. So here you can see that how the alternate contraction and relaxation of the ventricles, it results in sealing of the atrioventricular orifices by the atrioventricular valves. Now we will examine heart's tissue. The tissue of the heart, it performs the functions which are required to keep the double pump working strongly and steadily. Like other hollow organs, the heart is made up of layers of endothelial and connective tissues. So there are three layers which are called pericardium, myocardium, and endocardium. So we will start with this pericardium. Actually, the visceral layer of the serous pericardium, that is called epicardium, and it is a conical sac of fibrous tissue that closely envelopes the myocardium and surrounds the roots of major blood vessels. The epicardium, it excretes pericardial fluid into the pericardial cavity, which lubricates the tissue as the heart beats. So what is pericardial cavity? It is a fluid-filled space between the epicardium and the parietal layer of the serous pericardium. And the presence of the fluid, it reduces friction between the pericardial membranes. Now, there is a parietal layer of the serous pericardium, which is the serous membrane that is attached to the outermost layer of the heart, the fibrous pericardium. The fibrous pericardium, it is a thick white sheet of fibrous connective tissue that anchors the heart and the major blood vessels, including the aorta, to the sternum and the diaphragm. So what I want to say is that your heart is not just floating in your chest. The parietal layer of the serous pericardium, it also excretes pericardial fluid into the pericardial cavity. Now we move on to the middle layer, which is the myocardium. It is the thick muscular layer of your heart The myocardium, which literally means heart muscle, it is composed of cardiac muscle fibers that contract in a coordinated way to pump the blood out of the heart and into the aorta with enough force to carry it through the arterial system and out into the capillaries. Moving on to endocardium, it's a layer of endothelial tissue that lines the inside of the chambers. This layer is continuous with the vascular endothelium. Let's summarize. The pericardium, this is the outermost layer, and it is made up of two sacs. The outer sac, the fibrous pericardium. Now this outer sac, it consists of fibrous tissue, and then there is an inner one, which is the serous pericardium, and this consists of a continuous double layer of serous membrane. The fibrous pericardium, it is continuous with the tunica adventitia of the great blood vessels above, and it is adherent to the diaphragm below. It is inelastic. Fibrous nature prevents the over distension of the heart. Okay, so that was the outermost layer. Now there is the myocardium. The myocardium, it is composed of a specialized cardiac muscle which is found only in the heart.
So, dear students, after knowing the structure of cardiac muscle and the unique features of heart, you can see here the similarities between skeletal and cardiac muscle. The first similarity is that both skeletal and cardiac muscle are highly organized and they have aligned sarcomeres. So because of the organized sarcomeres, there are microscopic striations, or you can say there is seen a bending pattern under polarized light. The second similarity which you see in skeletal and cardiac muscle is that same essential principles and the molecular components of contraction, they are involved in the process of contraction. And here you can compare and contrast different features of skeletal muscle with the cardiac muscle, keeping this table in mind. You can look at the differences between skeletal and cardiac muscle. So the first difference between skeletal and cardiac muscle is that skeletal muscle is under voluntary control while the cardiac muscle functions autonomously. The skeletal muscle fibers, they contract independently of each other, each responding to individualized commands from the motor cortex. By contrast, cardiac muscle functions independently of somatic motor control, and the fibers are extensively interconnected so as to form a single functional unit. Now, the second difference between skeletal and cardiac muscle is the regulation of contractile force. Myocardial excitation always involves all the fibers, and there is no option of recruiting motor units to increase the overall contractile force as there is in the skeletal muscle.
Now, coming to the third difference between the skeletal and cardiac muscle, you see, the impetus of cardiac muscle contraction, it comes from within the musculature itself, unlike the skeletal muscle. A specialized pacemaker periodically generates action potentials that spread from myocyte to myocyte to involve the entire heart. The autonomic nervous system regulates the timing and force of contraction but does not initiate it. Repeat again, the autonomic nervous system, it does not initiate the contraction. Now here we have got some structural differentiation of heart, which include the troponin, the communication pathways, and transverse tubules and sarcoplasmic reticulum, and we are going to discuss it one by one, but only briefly. Troponin, it's a calcium sensitive heterotrimer which is associated with actin filament. In skeletal muscle, troponin must bind two calcium ions before the contraction can begin, whereas in cardiac muscle, only one calcium ion is required for the contraction. In the cardiac muscle, the pacemaker cells, they periodically generate spikes that spread from myocyte to myocyte until every fiber within the organ is involved. And this communication pathway that is made possible by the gap junctions and extensive cellular branching. And branching of the myocytes, it maximizes the interactions with adjacent cells. For electromechanical transduction, the action potential from cardiac pacemaker is conveyed to the core of the muscle fiber by T tubules and then to the sarcoplasmic reticulum. There are fewer yet wider T tubules which branch less extensively. Wonder what is the energy source of cardiac muscle? Cardiac muscle relies on aerobic pathways to supply ATP needed for contraction. The limited anaerobic capability makes heart muscle vulnerable to decreasing oxygen availability during interruptions in blood supply. And prolonged oxygen deprivations, the deprivation for minutes, it causes irreversible hypoxic muscle damage and myocardial infarction. So how the contractility, it is regulated in cardiac muscle? The molecular mechanisms underlying contraction, they are essentially the same in skeletal and cardiac muscle. But the key difference is the degree to which the sarcoplasmic calcium ions rises during excitation, because the calcium ions availability is used to regulate contractility in the cardiac myocytes. Well, the cardiac muscle, it is designed to take advantage of the relationship between the sarcomere length and force generation. And the advantage is to match its performance to the volume of the blood entering its chambers. So what does this Frank-Starling law of heart says? That increasing the sarcomeric length increases the force of contraction. As a result, it provides a way for the heart to increase the contractility when required to expel increased filling volumes. Well, the interesting bit about this Frank-Starling law of heart is that heart is constructed in such a way that in an empty heart, the sarcomeric length, it is minimal, and the possibility for further shortening and force development is small. In this figure, basically you are looking at the concept map for the cardiac function. You see, heart is a multi-chambered organ comprising the cardiac muscle. So the muscle contracts, chamber diameter decreases, and blood is forced under pressure from chamber to chamber and then out in the vasculature. Here I have got 15 points which you need to memorize about the contractile cardiac muscle. This muscle is located in the heart only. When cardiac muscle pumps blood out of heart, the cardiac muscle contracts according to sliding filament theory, and it is innervated by autonomic nerves. The cardiac muscle, it is under involuntary control, and hormones, they also modify the contraction. It uses ATP for contraction. It contracts as a syncytium. And keep in mind that in the cardiac muscle, the myosin ATPase activity and the speed of contraction is slow. Cardiac muscle obeys all or none law. When we say that autonomic nerves can modify the contractions, it means that this innervation can excite or inhibit the contractions. Then the gap junctions, as you can see in this figure, the gap junctions, they are present between adjacent muscle fibers at the intercalated discs. And contraction is initiated by the myogenic pacemaker potential. This is a very important point, that the contraction in the cardiac muscle, this is initiated by myogenic pacemaker potential. Calcium ions, they regulate the troponin in thin filaments that physically repositions the troponin-tropomyosin complex. And finally, our cardiac muscle, it obeys Frank-Starling's law. It means that strength of the muscle contraction, it depends on the initial length of the muscle fiber. And initial length is determined by the extent of filling of the heart chambers. So look closely at this figure and you can understand that the strength of the muscle contraction, that also depends on the entry of the calcium ions of the extracellular fluid into the muscle under influence of nerves or local metabolites and hormones.